Well, thank you. Thank you for going through this with us. Dr. Miyamoto, I know that you spent a lot of time, studying and looking for non-invasive methods of detecting rejection. You're actually funded from Enduring Hearts in our Transplant Longevity Award to look at some of this. So can you talk to us and take us now into some of the research that's evolving and what we can look forward to seeing maybe so children don't have to go through and get an angiogram and to detect CAV?
Sure. So I think to emphasize some of what Scott just talked about is that one of the hopes that we have as a field is that if we can identify CAV earlier, can we then institute treatments to either slow the progression or potentially even reverse the progression of CAV? And so that's sort of one of there are a couple of ideas here with looking at non-invasive ways of screening for CAV. And we know that at the present time, we don't have a reliable non-invasive biomarker or blood test that can be used to diagnose and risk stratify children that have had a heart transplant with CAV. So there's no blood test that we can use currently that will tell us if vasculopathy is present. And because of that, we are all reliant on these more invasive procedures like a cardiac cath or imaging procedures like a CT scan, which involves still some radiation and is obviously still a little bit stressful for the children. So the goal of the study that we did here is that we're trying to determine if we can find a blood marker that might help us determine the best timing for when to do the cardiac catheterization. We want to find a blood marker that might help us identify CAV really early in its course, maybe even before it can be seen with an angiogram. And then third, and hopefully, this is something that can eventually really be utilized, is can we identify new treatments? So we don't have great treatments for CAV right now. Dr. Auerbach talked about statins and the mTOR inhibitors like sirolimus and everolimus, and those have potential, but are there even more directed therapies that we can develop based on what we see in the blood? So the idea of a blood test or a biomarker is that you can draw blood on a patient just like is done for looking at levels and white blood cell counts and things like that. So in a usual lab, we can draw blood and then we can measure things within the blood. And there's lots of different characteristics that we can measure within the blood that might give us signals as to what is happening with the transplanted heart. So blood biomarkers can be proteins, they can be RNA, it can be DNA, all these little tiny pieces that can be detected in the blood of children who've had a heart transplant. And when we get the results back from these various biomarkers, we can then potentially determine whether or not the results of these biomarkers can be used to inform our clinical decision-making. So in this case, can we look at a biomarker in the blood and help us determine whether or not a cath is necessary because we're worried a patient has developed CAV, or the reverse. Maybe a patient's blood biomarkers are encouraging and suggest that CAV is not developing and we can avoid doing an unnecessary heart cath. And then that third tier there is, can we personalize therapy for kids so that some of these biomarkers might help us develop new treatments and new therapies for CAV. That is the holy grail and something we're certainly hoping to identify. So I'm going to tell you about two different studies we did. They're all based on the same types of patients or the same patient cohort. So we had 89 heart transplant patients at Children's Hospital Colorado that we had blood stored on and banked on. And we were able to, in these 89 patients, divide them into children who had CAV and who did not have CAV. And then we looked in the blood at something called microRNAs. And we'll talk a little bit more about this, but these are tiny pieces of RNA. Now, generally, RNA is turned into protein in the blood. So RNA is a piece of RNA that eventually becomes protein. But microRNAs are so tiny that they never actually get turned into protein. And so in this case, we're looking at these little tiny pieces of RNA to see whether or not they might be a biomarker of CAV.
A lot of acronyms.
So like I said, we did two different kinds of studies on these kids. So in the first study, we drew blood only from children who did not have CAV. So we took blood on patients that we knew did not have coronary vasculopathy, and we measured the microRNAs in the blood, and then we performed a heart cath 2 years after the blood draw, and we determined which of those patients still didn't have CAV and which of those patients did have CAV. So that was one study.
And you compared it to the level of the microRNAs?
So basically what we did is we measured the microRNAs in the blood of all the patients. And then we did this heart cath, and we divided the patients into those that did and didn't have CAV. And we said, okay, these microRNAs, looked at all the microRNAs that were evaluated, and we were able to pick out microRNAs that could distinguish patients who did and did not have CAV. But the important part about this study, and I'll show you the results, is that none of the children had CAV when we drew their blood. Okay, so nobody had CAV. We measured the microRNAs at that time point, and we're able to use those microRNAs to predict which patients would go on to develop vasculopathy versus those patients who were able to remain free from CAV.
So it was there before it actually presented himself.
That's right. The microRNAs. That's right. The microRNAs were kind of a signal that CAV was developing in the future.
That's way ahead of any kind of presentation that you could see in an angiogram, potentially.
That's correct.
You could get ahead of it if this worked, right? Okay.
That's correct. So the other study that we did is we drew blood on patients at the time that they were having a heart cath, and we're able to look at, did microRNAs correlate with the presence of CAV at the time of the heart cath? So this was not a predictive study. This was a study that just said, okay, in a child with CAV, do they have a different profile of microRNAs in their blood than those patients who did not have CAV? So this was not predictive. This was more just what microRNAs are present in the blood of children with and without CAV, and are they different? So those were the two studies. One was predictive, and one was looking at what these were different between the two groups known to have CAV. So this is what microRNAs are. So DNA is that's what makes up our genes, and that's a database of instructions. RNA is much more specific than DNA, and each RNA is associated with a specific protein. And you can think of protein as the functional product. So you think about RNAs as being kind of the instructions or the recipe, and the protein being the actual product, or for example, if you have a recipe, the protein would be the food that results from you performing that recipe. MicroRNAs are tiny little pieces of RNA that never make it into a protein. Instead, what they do is they kind of inform what happens with the instructions. So they inform RNA, they can inhibit RNA from getting turned into protein, but the microRNAs themselves are just regulators. They're not actually turned into protein. But in our lab, we have the ability to measure 754 different types of microRNAs that are circulating in the blood. And these microRNAs can give us clues as to the development of CAV.
So this may sound silly, but are they always present and
Yes.
there? Okay.
Yes, microRNAs are always present in the blood of transplant patients and non-transplant patients. All of us have microRNAs circulating in our blood, but the type of microRNA, there are 754 different forms of microRNAs that we can detect, and the level or the amount of each type of microRNA varies from person to person and can be used as a biomarker to help us identify diseases such as CAV and like Carolyn said, rejection as well as something we're interested in.
So the level of the microRNA that's signaling potentially CAV. Do we have a feeling from the work that you've done how early that is showing up in the blood?
Well, based on one of the studies that we did, it looks like even up to 2 years before the development of CAV, we see a change in the microRNA pattern. So it could be that even 2 years before a child develops CAV, their pattern of microRNAs in their blood may be changing.
That's so significant when you think about, potentially changing or intervening at that point.
That's right. I mean, that would be the idea is if we can know, 1 to 2 years before a child develops CAV, are there treatments we can institute that early that might prevent the development of CAV and alter that microRNA profile so that now the child does not go on to develop CAV? That would be the idea.